Well, hello everyone, and welcome back to Human Biology here at Chaminade University. Today we will be discussing DNA structure and function. This here is what's called the central dogma of gene expression, namely that DNA, which resides in the nucleus, creates RNA, which still is in the nucleus but is able to be transported out of the nucleus into the cytoplasm where it makes protein. So DNA makes RNA, RNA leaves the nucleus and makes protein in the cytoplasm. The process of making RNA from DNA is called transcription. Well, let me start over. The process of making DNA from DNA is called replication. The process of making RNA from DNA is called transcription, and the process of making protein from RNA is called translation. And I like to think of this as the fact that DNA and RNA are both in the same language, namely that of nucleic acids, so you're transcribing from within the same language. And when you're going from RNA to protein, you're translating it into a new language, namely that of amino acids. Okay, so both DNA and RNA are made of nucleic acids, and nucleic acids are very long polymers that are made up of nucleotides, so monomers and nucleotide. And each nucleotide has three major parts. It's got the sugar, which is either ribose or deoxyribose, okay, right, RNA or DNA, right, a phosphate group, and a nitrogen-containing base, which is an organic group. And there are 5 nucleotides, right, A, T, C, G, and U. In RNA, U takes the place of T. So for DNA, it's A, T, C, and G. For RNA, it's A, U, G, and C. Okay, so what do DNA and RNA stand for? Well, DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. The major differences between DNA and RNA, well, there are 3 major differences, and these are basically the questions that the gate guard will ask when the RNA is trying to leave the nucleus through the nuclear pore. So the nuclear pore guard is trained to decide whether or not the molecule that's trying to leave has uracil or thymine. If it has T, it must stay. If it has U, it's allowed to pass. It also will decide if it's double-stranded or single-stranded. A single-stranded molecule is RNA, double-stranded molecule is DNA. And lastly, it'll ask what sugar it's using. If it is using ribose, it's going to be RNA, and if it's using deoxyribose, it's DNA. As long as all 3 of these parameters are met, the strand of RNA will be allowed to exit out the nuclear pore into the cytoplasm, and that will be where the translation of that RNA into a protein molecule will take place. Now, DNA is a double helix. It's also known as a twisted ladder, right? And the way that the base pairs line up are very specific based on size and charge. That means that adenosine, or A, can only pair with thymine, or T, and cytosine, C, only pairs with guanine, G. And it's easy to remember that because A and T both have straight lines, and G and C both have curved lines. So anytime that you see A binding, you know it's T. Anytime it has C binding, you know it's G. And that actually will help us out when we get to Chargaff's rule, because if we know what G is, say G is 10%, we know C is also 10%. That means 20% is taken up, 80% is left for these two. That must be 40 and 40. So we'll talk about that in a second. But the fact that these only line up in these particular ways is really helpful in terms of determining the composition of the DNA. All right, so what holds the bonds of the base pair together is known as a hydrogen bond. So basically, if you were looking at this as a twisted ladder, your hands would be on the sugar phosphate, sugar phosphate, sugar phosphate backbone, and your feet would be on the rungs, right? That would be the hydrogen bonds between the 2 base pairs. Again, the sugar phosphate backbone, that's where your hands would be, those are going to be put together with the phosphodiester bonds. So this is what the DNA nucleotides look like. Here's the 4 nucleotides in DNA. You don't need to memorize this, don't worry. In fact, you won't even have to really know the difference. All you really need to know is that A binds with T and G binds with C. But I'll help you out here because, again, it's by size and charge. So as you can see, A and G are both bigger sizes, right? And T and C are both smaller sizes. So if we had 2 large together, we would have overlap, and if we had 2 small together, they wouldn't reach. So we must have 1 large and 1 small in order to have the correct size. And on top of that, you might not be able to see this, but A can only form 2 sets of hydrogen bonds here and here. Thymine can also form 2 sets of hydrogen bonds. Guanine can form 3, 1, 2, and 3, and so can cysteine, 1, 2, and 3. So it's by size and charge. This is large with 2 bonds, large with 3 bonds, small with 2 bonds, small with 3 bonds. So you have to have the large 2 with the small 2 and the large 3 with the small 3. Okay? I hope that kind of makes sense there. Anyway, the structure of DNA is really vital to its function because knowing whether or not you have A on one side means that you know for sure you have T on the other side. So that means that when we unzip the DNA, each strand has all the information that it needs to be able to be copied into a new set. So that means that the DNA actually has two exact copies of the information, which means that the information can be faithfully and accurately replicated to be passed on to the next generation. All right, so nucleotides are going to have different bases, right? A, T, C, and G. And if they have the large bases, right, namely A and G, they're going to be called purines because they have two rings. And the smaller bases are called pyrimidines, and they have a single ring structure, again, namely C and T. And there was a scientist named Chargaff that noted that the DNA molecules always have equal amounts of purines and pyrimidines. Always have equal amounts of one large and one small. And on top of that, we then learned that A always binds with T and C always binds with G. And so we now know that the amount of A is always the same amount as the amount of T, and the amount of C is always the amount of G. So if we know that we're aiming for 100, given one amount, we know any of the rest, right? So again, if we know A is 20, that means T is also 20. That's 40% out of 100. That means we have 60% left. So that would leave 30 and 30, right? So by knowing any one of them, you know the composition of the others. Again, purines are going to be these double ring structures here. That's adenine and guanine. And the pyrimidines are going to be the smaller ones, these single ring structures. And we must have one large and one small, so one purine and one pyrimidine for each of our connections, as well as the amount of base pairs must line up. So this can have two, not base pairs, the amount of hydrogen bonding must line up. So we can have 2 hydrogen bonds here and 3 hydrogen bonds here, allowing for the correct base pairing. All right, so on ribose and deoxyribose, they're each going to have 5 carbons, right? Because it's a 5-carbon sugar, and they're labeled 1 through 5. 1, 2, 3, 4, and 5. And again, 1, 2, 3, 4, and 5. And the major difference between ribose and deoxyribose is the amount of this little oxygen here. If this oxygen has been taken off, deoxy, removed the oxy, this one still has the oxy, so it's just called ribose. This is called deoxyribose because it's missing the oxygen. And that's really the difference, that one little oxygen molecule. One little oxygen atom, I mean. All right, so here's a DNA. Here's a double structure helix of the DNA. So we're going to have it condensed down into, again, we're going to have a twisted ladder. And if we're looking at where your hands would be on the ladder, if you were climbing the ladder, that's going to be the sugar, phosphate, sugar, phosphate, sugar, phosphate backbone. And if we're looking at the base pairs, again, C and G have 3 hydrogen bonds, A and T have 2, C and G have 3, A and T have 2. But if you were to put 2 G's together, they would overlap. Or if you would put 2 C's together, they'd be too far apart. So we have to have one large and one small, as well as matching, so it's by size as well as by charge, right? Okay, and in this way, because it's all held together by hydrogen bonds, we can actually just pull it apart very easily like Velcro. Just start ripping this end and ripping that end through a particular end, the unwinding mechanism. And then we can figure out that we can make a copy of it. So if we pull this one apart, we can make an exact replication of DNA, knowing that this is C, we have to bind a G on the other side. So each one of them has all of the information that it needs to be able to create the new molecule. Okay, so we start off with the nitrogenous base, and when we add the sugar, it becomes a nucleoside. We very rarely refer to things as nucleosides because we always have that phosphate group popped on there, which makes us a nucleotide. So the nucleotide is going to be the sugar, the nitrogenous base, and the phosphate group added on. ATP is a nucleoside triphosphate. Why do we call it a nucleoside? Because it does not have it does not have everything that we would need to call it a nucleotide. And it's produced in all cells, and it's the primary source of energy, as I mentioned, for cellular functions. We also can use GTP. So remember we had A, T, C, and G. ATP is used, so GTC, A, T, C, and G are all different, as I just mentioned, bases that we can use in DNA, but A is a base that can also be used as a triphosphate form for energy. And sometimes guanine, or GTP, can also be used as energy in cells, but it's not going to be the primary currency. We don't talk about it very often. All of the nucleosides in our body can be made into nucleotides for DNA replication if we get it from our diet, right? So we're going to be getting it basically from the things that we eat around us, and also conversion of other biomolecules. And we can use it for either DNA replication or to transcribe RNA from DNA. All plants and all animals have DNA and RNA, and we have specific enzymes in our digestive tract, so that when we eat other animals' DNA and RNA, we break down the nucleic acids into individual nucleosides, which then we comprise into our nucleotides. Because while we can consume the cow DNA, we can't just take cow DNA and turn it into human DNA. We have to break it down into the nucleotide components and then rebuild our DNA from the ground up. Okay, so change topics a little bit here. Let's talk about something called a telomere. It's going to be a repetitive region at the end of every chromosome, and it's basically a region that's meant to be degraded every time that this chromosome is replicated. So it's going to be there as a prevention for deterioration of the real message. So telomeres are like erasers on the pencils that are meant to be degraded to protect the actual pencil itself. And they shorten every single time DNA is replicated, because if you were to picture, like, I don't know, crocheting, at the very end, you can't use the yarn anymore because you can't wrap it around. It's just too small. So you lose that little bit. Well, it's the same thing with the DNA replication mechanism. Every single time you get to the very end, it can no longer hold on to the last 10 nucleotides or so. So you use a very little bit. Lose a very little bit, which means they get shorter after every cell division. This is why cells have a lifespan, and it's why we get older. So the telomeric aging effect was first discovered by when we were cloning a sheep. You may remember Dolly. Dolly was a sheep clone that was taken from an adult sheep cell. So they took a sheep cell, they popped the nucleus out, and they popped it into an egg, and they tricked the egg into thinking that it was, fertilized, but it wasn't. And what they did not know about were telomeres, so they did not elongate the telomeres, which is something that happens normally during embryogenesis. And what ended up happening was Dolly ended up suffering from a lot of elderly-style diseases because she had telomeres that were already shortened by the time she was born. So she was born prematurely old, right? And she's the reason that we know about telomeres today. Okay, so some people have telomeres that shorten faster than normal, and sometimes this can lead to different degenerative diseases, such as Alzheimer's disease, if it affects your brain, or cancer, if it affects certain regions of your body. And because telomeres shorten with each cell division, if something causes more cells to divide, like cancer, it will also increase the rate of telomere shortening. So telomeres will get shorter and shorter. And eventually, once you reach through the rest of the eraser, you start degrading into the pencil. So eventually, once you degrade all of the telomere, you start degrading into the real good stuff in terms of the DNA. Some other things that can cause increased cell division include inflammation. And inflammation is also linked to degenerative diseases. And they think that that's also because of the fact that it causes telomere shortening. And high levels of stress have also been linked to telomeric shortening. Okay, so here's the DNA. Here's the chromosome, right? It's one chromosome. This is the centromere, which connects them together, right? So we have the short arm and the long arm, and at the end of these, we have the telomeres. Telomeres are repeat regions that are, I don't want to say nonsense, but they're non-coding regions, and they're literally just meant to be degraded. And again, as we are undergoing embryogenesis, they get elongated again in the early stages after fertilization. So here's the end of the coding region, if you want to call this like the pencil, right? Here's the telomeres. It's just going to have repeated AGGG, CCCAAT repeats over and over and over and over and over again, meant to be degraded each time the chromosomes replicated, right? So all eukaryotic chromosomes have these special telomeric sequences at the end of the chromosome, which are going to shorten with each DNA replication and going to get elongated again during the process of embryogenesis. Now, prokaryotes don't need this. Remember, their DNA is not going to be in the shape of the X. It's going to be circular DNA. And as I mentioned, during gametes or gametogenesis, right, during stem cells and germ cells, we're going to have an enzyme called telomerase, which is going to elongate the telomeres back to their original length. And this is the step that Dolly missed, right? Dolly did not undergo telomeric lengthening and therefore went into her life as though she had started off at the end of her life in terms of her telomere length. But cancer cells are also pretty tricky. They also produce telomerase, so we can use telomerase as a marker for cancer cells, because cancer cells are immortal, right? They don't die. They're going to grow over and over and over and over. They're never told to stop. And they also seem to be able to replace their telomeres. So we can't use a telomerase drug to prevent aging because we might end up giving someone cancers, right? Because you might end up producing immortal cells and increasing the rate of cancer. Okay, so here's just some fun DNA trivia. Average length of a single chromosome is 2 inches if it were all stretched out. Mind you, it's inside a microscopic cell, so it's condensed down into a teeny tiny coil. We each have 46 chromosomes in each cell. That's going to include 22 autosomal pairs and 1 sex chromosomal pair. The total length of DNA per cell is about 2.3 meters, and if we took the cell, all the DNA in our body, we would be able to reach the moon with it. Okay, so let's settle in for DNA replication. When we are copying DNA, We know that the two strands of DNA are complementary to each other, which means that they both have all the information that they need because they're mirror images of one another. And we also are able to pull it apart at those hydrogen bonds, which are going to be relatively weak and easy to separate. And so what will happen is we will separate it out, and given the sequence on 1, say this is the sequence that we have, we'll be able to pull in the opposite sequence. So if you were looking at this strand, you were asked the complementary strand, everywhere you saw a G, you would bring in a C. So it would be C C A T G G T C A, right? Okay, so this is what we were talking about with DNA replication. So here's our parental DNA. We're going to separate it out. And each strand, this purple strand and that purple strand, are both going to serve as templates for the newly synthesized strand, which is shown here in blue. Right? And then when we undergo the next set of replication again, we're going to separate back out again. So it's called semi-conservative replication because these two purple strands, once they separate out, they never come back together. They separate out, find new partners, and create a whole new line. Then they separate out again, create new partners, and create a whole new line. So every single new strand is made up of one original strand and one new strand. And this is important because that means that if there's a mismatch between the two and an enzyme that's supposed to be proofreading comes up and says, hey, you have a G here, you have a G here, but an A here, and that's a mismatch because G is supposed to match with C, but A is supposed to match with T. So which one is it? Well, it will always take the side of the older strand and cut out whatever is on the newer strand because it assumes that the older strand knows what it's talking about, which means that mutations that may arise in that older strand will become enduring for the next generations. That means passed on for the next generations. Okay, this here is talking about how bacterial DNA replicates. Bacterial DNA is going to replicate by, remember it's a circle. This is going to start at one point of origin and separate out. And the DNA is going to be replicated in both directions at once until we finally end up with two full circles. So they finally just separate off into two separate sets. Replication is going to begin at a point of origin all the time. In prokaryotes, there's one point of origin. In eukaryotes, there's many points of origin. And a specialty enzyme called helicase is going to open up the DNA by undoing those hydrogen bondings. The area where it's unwound is called the replication fork. And we have specialty proteins that are going to stabilize these separated strands of DNA so that DNA polymerase can come in, making the polymer, right, making a DNA polymer, moves along each strand of that DNA and adds the complementary nucleotides. If it sees a G on one side, it puts a C on the other, for example. Now, the DNA poly can only add new nucleotides to the existing strands, right? So the very beginning part, we have to use primase. So it can't DNA poly can't start a strand. So primase is going to start a strand. It's going to add a small fragment of RNA, so not a DNA, it's called an RNA primer, to the DNA. And then the RNA primer being complementary to the DNA will later on be replaced with DNA once the strand actually gets going. So once the whole thing gets boosted, that RNA will get cut off and DNA will get popped on. Now, in eukaryotes, like us humans, the DNA strands are what we call antiparallel. I may not have mentioned this yet. In fact, I'll go backwards a second. I'll go backwards several seconds here. As you can see, this one is 5'. So if you consider this to be like left and right or up and down, etc., 5' and 3' are opposites of each other. So this end here is 5', and we go all the way down, it becomes 3'. In this case, it's 3', and if we go all the way down, it becomes 5'. That means that the sugar phosphate order is opposite on the two of them. They're anti-parallel. They are not going in the same direction. They're going opposite directions. So sorry I didn't mention that previously. So since one strand is oriented 5' to 3' and the other strand is oriented 3' to 5', we have to put them on in different ways. So one strand called the leading strand is able to go in a straight line because nucleotides are only able to be added to the 3' end. But the other one's going to have a lagging strand, and it's only going to be able to be made in discontinuous segments. That's called Okazaki fragments. Basically, what happens is the polymerase can add them in the 5' to 3' direction, but since it's opening this way, but it's adding them this way, it'll be doing this, right? Be kind of jumping backwards to do it. Not sure if it's going to show me the Okazaki fragments. Yes, it is. Okay, good. So here's a DNA strand, right? We're unwinding in this direction. For this one, we can go no problem, right? In the leading strand, we're just going to follow because it's only adding in 3', right? No problem. But for this one, we're going to be adding 3' to 5' that direction and then have to go back here to add 3' to 5' that direction until we're constantly, so we're going to have to go back, start over, then go back again and start over. So the lagging strand is actually going to have to be synthesized in small segments, whereas the leading strand can just go straight on through because we can only add to the 3' ends for the DNA replication. Okay, so while we're having access to the DNA is going to have a little bit of tension relieved because normally the DNA is under a lot of tension. It's supercoiled, right? So in order to relieve the supercoils, we use an enzyme called topoisomerase, and that's used both in eukaryotes as well as prokaryotes. And it's also used in viruses to integrate viral DNA into whatever cell it's infecting. So it's kind of a sneaky enzyme that can be used to infect cells as well. Okay, so this application question is referring to viruses and cancer. And it states that viruses can cause DNA mutations that lead to cancer. Viruses instruct strands of DNA into the cell during infection. What type of DNA mutation can this cause? A point mutation or a recombinant mutation? All right, so let's talk about DNA mutations for a little bit so we can talk about which type of mutations we have. We haven't even gone over that yet, so it all seems so new. So DNA mutations occur when replication is done incompletely or erroneously when a mistake happens during replication. Now, we have a proofreading gene. Actually, we have a ton of proofreading genes. About 130 different genes code for DNA repair enzymes. So everything that is copied is going to be edited and edited and edited. So most of the time, any damages that are found are going to be fixed via a process called proofreading, where repair enzymes and polymerase, etc., are going to compare the new strand to the old strands. And when it says fix any incorrect nucleotides, they're taking the old strand's word for it every time. But proofreading is still not quite perfect, and we can have mistakes occurring where the DNA mutations arise. Again, a mutation is going to be an enduring change in the DNA itself. And what happens when DNA changes happen? Well, oftentimes we try to catch it. We use proofreading and repair enzymes to try to catch it, but sometimes we don't catch it. So sometimes mutations occur and it causes the cell to die, right? Programmed cell death, that's apoptosis. Or it causes the cell to look so different to its other cells that it's going to be killed by immune cells because it appears to be going awry. But sometimes mutations still endure, and we can end up with disease states, or in very, very infrequent cases, something that's going to be an allele, so a new variant of a particular type of disorder. Okay, so there are many ways to alter the genetic message. There are two main ways. The first is going to be a point mutation, where we are changing one nucleotide. Usually happens from an error in replication. It can be a substitution, an addition, or a deletion. But we can also have what's called a recombination mutation, where we can cause a position of the gene to change. We can cause a change in the position of the gene for either all of it or part of it. We can shuffle things around a little bit. And if we end up with a mismatch of approximately 1 in every 1 million base pairs or so, but we have a polymerase mechanism that causes a pause. If there's a mismatch, the polymerase will pause, and then something will happen, and it'll edit it out. And then the exonuclease section is generally going to the polymerase enzyme is going to take the wrong part out. That's called proofreading, or one set of proofreading. And then they remove the error right there on the site, and the correct nucleotides replace the mistake. This is the equivalent of you taking notes and realizing as you're writing the word that you're spelling it wrong and fixing it right then. But sometimes that word is spelled wrong, and you don't even see it when you're writing it, and then later on someone has to go through and fix it. So we have other repair enzymes that are going to be editors, basically, that go back and read and say things like, you wrote the twice, or whatever. Right? So repair enzymes can go back and correct DNA mutations as well. However, DNA mutations actually are very beneficial in terms of evolutionarily speaking. So within a population of organisms, having a variety within the genetic makeup means that some individuals are able to arise to the challenge no matter what the environmental challenge is. However, most DNA mutations are not beneficial. So some of them can be, but a lot of them are not going to be beneficial. So in a specific organism, they can be either beneficial or what we call deleterious, which would be bad for that individual. Now, we know that there's a lot of chemicals and substances, like UV light isn't a chemical, but is a substance, that causes an increase in the rate of DNA mutations, and we call those mutagens. And we also have a word for a substance that causes mutations in DNA and also causes cancer, and those are called carcinogens. Now, mutations generally arise randomly, and because of that, most mutations are detrimental. Picture that most typos are probably not going to be good for your storyline, right? But if you accidentally were, like, say you were writing the fat cat and you accidentally wrote the fit cat, and all of a sudden you went, that sounds much better, and I'm going to change the whole cat to be fit through this whole story. Well, that's evolution, right? Now you've changed something for what you consider to be better, right? So sometimes mutations can change for the better, but generally, because they occur randomly, most of them are detrimental or deleterious. So mutations can arise in two different cell lines, either in your germ cells or in your somatic cells. In your germ cells, like your sperm and egg, these are going to be what we call enduring mutations. They're going to be passed on to future generations. They're going to be really important for selective pressure, evolutionarily speaking. And we also have mutations in somatic cells. Say, for example, my grandfather smokes a pack of cigarettes a day and ends up dying from lung cancer. That's not passed on to future generations because it was in his lungs and not in his testes, plus I was already born. So they're passed to other somatic cells derived from them, as in any lung cells that he made from those cancerous cells would also be cancerous cells, but my lungs wouldn't have cancer in them, knock on wood, right? So anything that's in your somatic cell line is not passed on to future generations, but is passed on within the organism to any of the cells that are derived from that line. Okay, this here is just a series of different types of mutations that we can have occurring. Here is a missense mutation. Here, and the second line here, and this one here is going to be a frameshift mutation. But before I jump into this, let me orient you to what you're looking at. You're looking at the DNA sequence here, 5' to 3', 3' to 5'. That's going to get translated into mRNA. And then mRNA, every triplet is going to get turned into, so this AUG gets turned into methionine, ACC gets turned into THR. Each one of these is going to become an amino acid. So in the polypeptide chain, you're going to have in blue the amino acids that are going to be in there. And if you can see here that this mistake here, going from a C to a G, is going to give us a C here. So the altered sense codon is going to give us proline right here instead of serine. So it's only going to change one amino acid, and the rest of the sequence is going to remain the same because we only changed one nucleotide. But here we have what's called a frameshift because we've inserted a base pair. And so by inserting it, we've made everything downstream off by one. So previously it would have been GGT, but now it's GAG, and then everything else downstream gets pushed over one, which means that we have now entirely amino acid changed everywhere downstream. That's called a frameshift. Sometimes it can lead to a truncated protein or a nonsense protein. Very occasionally it leads to a protein that's helpful, but very, very, very infrequently. Usually these mutations are going to be what we call deleterious or bad for the organism. All right, so again, we can have multiple different types of DNA mutations, including substitution changes where we are changing out one base pair for another. We can have an insertion or a deletion where we are adding or removing a base. And again, a consequence of deletion or addition would be a frameshift, right? So everything downstream of that would be off, unless we inserted or deleted in sets of 3. So if you inserted 3 or 6, you would just add an amino acid in the middle or 2 amino acids, and then downstream would be normal again. But if it was not a set of 3, you're going to result in a frameshift mutation. And again, the frameshift mutation is the most deleterious or detrimental because the final protein is completely drastically different or might not be made at all. So not only are you losing protein function, but you may be gaining a totally aberrant protein function. And so here's an example. If we start off with the cat saw the dog, a substitution would be something like the bat saw the dog. Like that's what I said earlier. The fat cat ran, the fit cat ran. You still get the gist. It's a little bit different, but everything downstream is still the same. But if we do an insertion or a deletion, now we have the right? Nonsense. It's not even English anymore. And because we can't interpret anything else, we're literally just going to be making whatever amino acid this calls for, and it's going to be nonsense. So that's what insertion and deletions lead to, how insertions and deletions can lead to frame shifts. All right, so we also have transposable elements. So these are jumping genes. A woman named Barbara McClintock was thought to be totally crazy, and it turns out she was just a genius. But she found regions of DNA they were able to move within a cell, basically like copy and pasting. So these are jumping genes that are going to be really important in variation and evolution, but sometimes they can be detrimental to an organism depending on where they leap. Okay? So if, for example, if a mutagen caused the sugar phosphate backbone to be broken, the cell will try to fix this by adding the DNA fragment to another piece of DNA, right? And this is how we can fix broken DNA, but can also cause segments of DNA to be popped to different chromosomes. Like if you were to try to to graft something you have a burn, so you want to do a skin graft. All of a sudden, the skin, now, yes, you do have skin on the area that you did not have skin on before, but that skin came from your thigh, so it's been moved around, right? So what would happen if this occurred during germ cells? Well, it might affect where things lined up during meiosis. All right, so this is what we're talking about here. Here's a normal gene here that has 2 regulatory sequences shown in yellow on either side. And here's a mutated gene that has normal regulatory sequences on either side, but this middle part here makes irregular or aberrant gene products. All right? So this would say if a transposon jumped in here, or if a transposon jumped upstream, we'd have regulatory sequences might be mutated. And although the functional product would be transcribed, it might not be transcribed at the right amount. It might be too much or too little. If the jumping gene or transposon ended up downstream, this would be a silent mutation where nothing would happen. So depending on where the transposition jumps in, right? It's like having something interrupt your movie. It might come in the previews, in which case it might be a little bit of annoyance, or it might come in the middle and completely disrupt what's happening in the central plot line, or it might happen well after you've left in the previews, or sorry, in the what do you call it when all the names are aligned at the end? I forget what that's called. The credits. All right, anyway, I'm off topic. But the point is that depending on where the transposon leaps in, it may have a big effect, a minor effect, or no effect at all. And the primary source of new alleles, or just basically what makes the difference between all the phenotypic or outward appearance variability in a population, is due to non-detrimental DNA mutations. So here's an example of non-detrimental DNA mutations in an organism called Drosophila, right? So Drosophila, we can have vestigial wings, which are really tiny, right? We can have cut wings, which are kind of bent and folded, curly wings, which are curled under, rudimentary wings, which are all the way underneath. What's called a rotated abdomen, so the abdomen pulls under. Diachetes, so the wings go all the way out instead of folding down normally. So there's a lot of different things. So this is just for wings. We could also have eyes. So here's the wild type with regular eyes. Here's the bar eyes that are abnormal. Wild type with the regular abdomen or the abnormal abdomen. So depending on where the DNA mutations end up, they can cause a difference in the phenotype or outward appearance of the organism. All right. So within our DNA sequence, we have introns and exons. You can picture this like a commercial break in between your TV series. So exons are sequences that code for mRNA that will go into the protein, the actual TV running time. And introns are removed. These are the commercials that you're going to cut out if you were going to be taping the episode, which I know really dates me and nobody actually uses TiVo anymore. But if you wanted to stop it so that you were making it that you didn't have to watch the commercials later, you would be cutting those out. Those are the introns. Right? And if a mutation occurs in an intron, which is going to be spliced out anyway, there's not going to be any effect on the protein. That's called a silent mutation. So let me go back a second just to show you what I mean by that. So here, if we're looking at a gene, come on. We're looking at a gene. Here's the regulatory. Here's the start and the stop. That's what I mean by regulatory sequences. So start. And then this region would be the exon. This is what we're going to be putting together. All the green will end up becoming a protein. And these guys will be cut out as introns and not actually utilized to create the protein. So if we were to put that transposon in just this gray area, we might not have any problem at all because that'll end up being edited out in the long run. So that's just what I mean by introns and exons. Sometimes we can have mutations occur in a base that doesn't affect the protein sequence at all, and that's because there's a little bit of wiggle room in what we code for, what we call the codon dictionary. So sometimes, in fact, multiple different sets of 3 will code for the same amino acid. So if it's not a huge change, sometimes you'll be coding for the same amino acid, and that'll also be considered a silent mutation. Okay, inside our body, we have natural defense mechanisms that are called natural killer cells, and they're always on the lookout for cancer cells or cells that are growing abnormally, cells that are taking up more space than they're supposed to, cells that are growing awry, etc. So natural killer cells that have cancer cells, for example, that have brought in more blood supply than they need or have started to extravate, move along. So cancer cells start doing abnormal things, and these natural killer cells are kind of like the police, and they start recognizing the chips and trademarks of cancer cells and will attack them early on. And that was just an example of what one looks like. Now, here are some examples of genetic diseases. Cystic fibrosis is one, Huntington's disease is one, sickle cell anemia, and cancer. These are all different genetic diseases that arise based on a mismatch in genes that do something important. So in cystic fibrosis, in a gene that's coding for the chloride channels, and during respiration. So it's going to be able to, they're making thicker mucus than normal and aren't able to expel it. So they're having a hard time breathing. In Huntington's disease, we have an insertion of CAG repeats in a chromosome that is going to lead to excessive shaking and eventually death. And this is an adult onset disorder. So this is kind of sad because usually this happens to someone after they've already had their children. So children who know their parents have had this disorder just kind of have to sit and wait. Although now we obviously have genetic testing and you can find out. Well, and you can also make sure that you test your embryos before you implant them so you can make sure that your offspring are free from this disease, which is nice. All right, so sickle cell anemia is another example of a mismatch, in this case in the hemoglobin gene. Cancer usually has multiple different genes that have gone wrong, so genes that are involved in the cell cycle, so either oncogenes or tumor suppressor genes. So you can picture this like the gas on the bus and the brakes on the bus. Either one can go wrong. If you're having a problem with the bus speed, right, if you have too much gas, the oncogene can overtake it, or you can have a tumor suppressor gene that just isn't working, so you just don't have brakes. Either one, you can end up with a cancerous activity because the cell cycle is continuing to be unchecked. So when I say you don't have brakes, it means the cell cycle is never being stopped. It's just on all the time, right? Here's another couple examples of genetic diseases like PKU and colorectal cancer. But let's talk about how we get DNA mutations. They can be acquired during DNA replication, during transposition, like we talked about with the jumping genes. They can be inherited from one of your parents. Approximately 10% of diseases are this way. They can arise from mutagens and carcinogens, things like lung cancer, etc., usually come from external environmental causes. Or we can acquire DNA mutations from viruses like human papillomavirus and its connection to cervical cancer, for example. Now, we also have things that are called, that are going to be external sources. So not necessarily mutations themselves, but things that cause mutations, like radiation and chemicals. So some forms of radiation include UV light and X-rays that are involved in the creation of skin cancers sometimes. Some other chemicals can include pesticides, herbicides, industrial chemicals, pollution, food chemicals, different drugs people might have put into their body, etc., as well as viruses. We talked about the human papillomavirus, which causes warts, and these warts can cause cervical cancer. HIV is human immunodeficiency virus, which is going to attack the T cells. When it becomes full-blown, it becomes acquired immune deficiency syndrome, which can cause sarcoma and blood growth problems, etc. So basically, no one really dies of AIDS. They die of secondary complications because their immune system is unable to fight off simpler disorders, right? So it's acquired immune deficiency syndrome. And these days, we is actually really looking positive for victims of HIV and AIDS, and that we have drugs that you take once a day that are relatively inexpensive pills that are antiretrovirals that can keep your viral load extremely low, in fact, so low that you can stop them from being passed on to your lovers and partners, which is fabulous compared to where we were in the 1980s. Also, we have hepatitis B and C. These are infections of the liver. Right, so we can have hepatitis infections, we can have alcoholic-induced hepatitis, or we can have hepatitis A, B, and C, which are viral infections, and B and C are known to cause liver cancer. We also have Epstein-Barr virus. Anybody ever heard of mono, or the sleeping sickness, the kissing disease? Mononucleosis is going to be contracted through saliva or salivary contact, and it can cause lymphoma in rare extreme cases. Okay, so this here is the answer to the application question. So when viruses that cause cancer infect human cells, they insert their DNA into the cell, and that DNA can be incorporated into the human DNA. So this is how recombinant mutations, because it involves a large sequence of DNA. When the virus DNA is inserted, this can sometimes cause insertional inactivation, which means the viral DNA interrupts a human gene and inactivates it. If the affected gene is one that is involved in the cell cycle, such as a tumor suppressor gene, this can lead to cancer. If the viral DNA inserts itself into a regulatory region of a gene, this can alter the regulation of the expression of the gene, with the result that too much or too little of that gene may be expressed and can also lead to cancer. If the viral DNA inserts into a region of the chromosome that does not contain genes or regulatory regions, the viral infection will not cause cancer. For example, some strains of human papillomavirus cause cervical cancer, but not every person infected with HPV will contract cancer. Thank you so very much for listening to my lecture today, and as always, happy studying.